simple Mendelian genetics, which many of you may have had before. Hopefully you'll still learn some new things. I want to remind you that you have a quiz in class on Monday, and the quiz will start on time. So you will need, I'm not going to wait until people are here. I'm going to start the quiz at the top of the hour. Okay, so please try and get here on time. Make sure you bring a pencil and an eraser. You might want to bring 2 pencils if you're nervous your pencil is going to break. A few of you guys forgot erasers last time. Please bring them. You'll be able to sit where you want, but you'll need to put all your stuff under your seat. The quiz will happen at the beginning of class, and then we'll have a lecture, as you did for quiz 2. This quiz will cover everything from mitosis through the end of the lecture today. So everything since the last midterm. Any questions about the quiz? We'll be giving you your Scantron again this time, just like last time. It is multiple choice. Any other questions? Okay. I think I'm too loud. So today we're going to talk about Mendel, and we need to start with a couple of key definitions. Genetics is full of vocabulary that you need to understand. So the first word we're going to introduce here is phenotype. The phenotype is the observable characteristics of an organism. Sometimes these are also biochemical characteristics that we might measure in the lab. Whereas the genotype refers to the genetic makeup. It is which alleles the individual has. So the genotype and the phenotype are not always the same. The genotype is referring to the genetic makeup, and the phenotype is the observable physical characteristics of the organism. We'll talk about this more later. They are different things. It's important to understand that Mendel's experiments worked because he began with true breeding varieties. So what does that mean? That means that these peas with the round phenotype, when they were self-pollinated for many generations, they produced only peas that looked like themselves. So they produced peas with the same phenotype. Those peas were true breeding. When he bred the wrinkled peas, self-pollinated them for many generations. He only got out wrinkled peas. So these are true breeding varieties. What that means in terms of genotype is that the peas that he started with were homozygous. They had 2 of the same allele. What are the F1 and F2? Well, the parental generation here, those are the true breeding parents that Mendel started with. So if you self-pollinated this purple flower for many generations, you get nothing but purple flowers. If you self-pollinated the white flower for many generations, you'd get nothing but white flowers. They're true breeding. When you cross those 2 true breeding strains, it's called hybridization, and what you get out are hybrids. These are called the F1 generation. When you self-pollinate or cross-pollinate, cross-breed if they won't self-pollinate, you end up, the F1 generation, when you cross it to itself, you end up with the F2 generation. So we'll be using those terms to describe parts of Mendel's experiments, and you should know what they mean. Now, as another example, this here is a true breeding Labrador. So it's a P generation. This is a true breeding poodle. And these were bred to produce my dog, Daisy, who is an F1 Labradoodle. So she's a cross between a Labrador and a poodle. 2 true breeding strains produce the hybrid. She is the F1 generation. This is something that we've touched on before too, but I want to make sure that you guys get this concept. We've talked about this favorite ice cream flavor locus that I made up to make some genetic points. We said that diploid individuals will have 2 alleles of this gene. In this case, the person has a chocolate allele and a strawberry allele. So each diploid person can only have 2 alleles. They will have 2 alleles. They could be the same or different. But in the population of everybody in this room, there could be many, many different alleles. You could have chocolate, strawberry, vanilla, chocolate chip, mint chip, pistachio, Terry Garcia, chunky monkey, whatever you want. And many different alleles are available in the population, but each diploid individual can only get 2 alleles. This is another thing that several students have come up to ask me about. How do you talk about this person who has 2 of the same alleles? They have 2 alleles, but those alleles are identical. They have 2 of the same allele. So they don't have 1 allele. They have 2 alleles that are the same. Each diploid individual can have 2 alleles that are the same or 2 that are different. Either one is okay. They still have 2 alleles. Now, maybe this will help you, maybe it won't, but a good analogy is socks. This is not my laundry, but my laundry looks a lot like this. Nobody likes to put away socks because it's so hard to find their mate. So this is a big pile of socks, right? If you walk up to this sock pile, you need 2 socks. So you're going to take 2 socks, 2 alleles out of the pile. The likelihood of getting 2 socks that match depends on the relative frequency of each kind of sock in this pile. So for me, in my house, I went to Target and bought my daughter 12 pairs of the same kind of white sock. So that whenever she walks up to the laundry pile, the probability of her pulling out 2 of the same allele is high. Okay, so I've altered the frequency of my sock alleles in my home laundry pool. And this is going to be a concept we're going to go back to, that the frequency of an allele in the population can help you figure out what the genotype of individuals are. We just said that diploid organisms have 2 alleles that can be the same or different. When we write these alleles, we use italics because we're talking about genes. When we're talking about genes, we italicize the letter or the word so that everybody understands we're talking about DNA, genes. When we're talking about the protein produced by the gene, we will use normal print. We already brought in the word homozygote. Those are people with 2 of the same, or organisms with 2 of the same alleles. There are 2 homozygotes here, homozygote dominant and homozygote recessive P. A heterozygote is going to have 2 different alleles, and there's only one of those here. This guy is the heterozygote. Now, your textbook uses the letter of the dominant allele to represent the gene. That's not always true, but that's what we're going to do in this class. So these P's are round. Round is dominant. We're going to use a big R for the dominant allele, because we use capitals for dominant, and a little r for the recessive allele of the round genes. Now, how do you determine which is the dominant allele? If one's capital, you already know. But if you don't know, how do you figure it out? Well, the phenotype of the heterozygote tells you which is the dominant allele. So our person who had a chocolate and a strawberry allele, if they like chocolate, chocolate ice cream is the dominant phenotype. If they like strawberry ice cream, then strawberry is the dominant allele. So the phenotype of the heterozygote tells you which is dominant. If strawberry is recessive, then it will take 2 copies of the strawberry allele for somebody to like strawberry ice cream. Now, Mendel used a whole bunch of different true breeding P's in his experiment, and this table shows you all the different traits that he looked at. And the way he figured out which trait was dominant was he looked at the F1s. So the F1s are the heterozygotes, they're the hybrids. Whichever of these 2 phenotypes was present in the F1 was the dominant phenotype. So here, if you cross a round P with a wrinkled P, the F1 is all round. So you know that round is the dominant phenotype. The dominant allele, as I said, is the one whose phenotype is present in the heterozygote. But how do you tell what the wild type allele is? How do you know what the wild type phenotype is? The phenotype that is most often observed in a natural population is considered to be the wild type. The wild type allele could be dominant, but it doesn't have to be. The wild type allele is the one that's most common in a natural population. So we're going to do our own experiment here in Bio 97 for the favorite ice cream flavor locus. We're going to do an experiment to figure out what the wild type allele is. What is your favorite ice cream flavor? Vanilla, chocolate, cookies and cream, mint chip, or coffee? I selected these flavors based on what Google tells me 20-somethings in California like. So I apologize if yours is not here. No, you guys answered this question faster than any other question. That's pretty easy. Okay, so I was surprised that it was cookies and cream, but A was just saying. So in the natural population of bio-93 students, the wild type allele is cookies and cream, apparently. We don't know if it's dominant or recessive. Okay, the wild type phenotype here is liking cookies and cream. So polydactyly. We can't cross undergraduates to find true breeding undergraduates to find out which ice cream flavor is actually dominant. So we're going to do this with polydactyly in cats. Polydactyly means that you have extra fingers and toes. And when I was a kid and took ballet class, the guy who did the lights for our ballet shows was polydactyl. He had 6 toes, his name on each foot. His name was Lester, and he never wore shoes because it was hard to get his feet into shoes, they were uncomfortable. And we were very impressed with his 6 toes. But cats are also often, or sometimes, polydactyl. These are pictures of a variety of polydactyl cats. Has anybody here ever seen a polydactyl cat? Can you raise your hand if you have? Okay, this is pretty similar to last time. Maybe there's 6 or 8 people who've seen a polydactyl cat. They're pretty rare, right? So polydactyly is dominant, but it's not the wild type allele. If you walk up to the sock pile, you're not going to take a polydactyly allele out very often because they're uncommon. They're rare. So you don't see a lot of polydactyl cats around. This is another question that people often find confusing. A mutation in the promoter of a gene could produce a recessive allele. Is that true or false? Did you have a question? I can't hear you. That's very true. Thank you for asking that. If an allele is common, that does not mean it's dominant. That is correct. Just because it's common doesn't mean it's dominant. You have to look at the heterozygote to figure out which is dominant. Okay, if you haven't answered this one, give your best guess. This is very similar to section A too. The answer is true. Okay, so why is it true? One of the TAs last time gave me a really good example for you guys. So you remember the purple and the white flowers earlier, and purple was dominant and white was recessive? Okay? The reason why those white flowers are white isn't because they make a white pigment. It's because they make no pigment. Because the promoter of that allele is messed up. So it doesn't even get expressed. So there's an example of a recessive mutation in a promoter. One thing that I think is confusing about this is people think that for alleles to be different, they have to be different in the coding sequence. But that's not true, right? I could make a mutation in the promoter. I could make a mutation in the 5' UTR. I gave an example to section A. What if I put a mutation in that added a new AUG upstream of the real start codon? So the ribosome scanned down and it started reading at that new AUG. It would make a completely messed up protein. Even though the coding, original coding sequence is not different, that allele is going to produce a very different phenotype, a different protein. I could make a mutation in the 3' UTR that destabilizes the mRNA. And now that mRNA will get degraded and the protein won't even be produced. So you can have recessive alleles or dominant alleles that have mutations in non-coding regions of the gene. Did that clarify that? Make it worse? Anybody want to ask a question? We're good? Okay, the answer is true. So now we're going to talk about the principle of segregation, one of Mendel's laws. This one is that alleles separate or segregate into gametes. I changed the wording on the slide a little bit to match your textbook better. I used to teach this with a different genetics textbook, so I just switched the wording to be more in keeping with your book. So what this means is that when an individual makes gametes, each gamete will get one. The alleles separate, one allele, and it's equally likely that a gamete will get either of the two alleles the diploid individual has. And you guys know this because you know a lot about meiosis that Mendel did not know. So let's say the big R allele is on the paternal chromosome, and the little r allele is on the maternal chromosome. These guys are going to separate, and at the end of meiosis, you're going to have only one allele in each of the four gametes. The way that I help myself separate, in my mind, the principle of segregation and the principle of independent assortment is that the principle of segregation is talking about one genetic locus. It's talking about the two alleles at one locus separating or segregating into different gametes. When we get to independent assortment, we'll talk about how it's different. So for the principle of segregation, it's one locus, two alleles separating into separate gametes. Okay, if that didn't help you remember it, I have another way that might be useful.
Oh, why, dear God, did you make it so complex to understand the offspring that result when there is sex? For there is one monk among us who can tell how it all works. We feel like jerks next to Greg or Mendel. The answer's in my garden where I planted different beans and sprinkled pollen as I pleased and counted out the progeny. What did you discover in your garden with your peas about those factors we can't see but which explain our family trees? Here's the news, they come in twos, they separate, except to fate. If an egg or a sperm has a trait that will dominate. Here's the news, they come in twos, they segregate, it's up to fate. If an egg or a sperm has a trait that will dominate. And when they join together, my forecasting's most impressive. Catch it 3 times out of 4, I'm right unless they're both recessive. To you all your success in counting in my green thumb, but where these hidden factors are, well, that I cannot fathom. Here's the news, they come in twos, they segregate, it's up to fate. If an egg or a sperm has a trait that will dominate, later on the world was on and what he learned from those bonds. But back then no one hurrah, Gregor Mendel, but he's gone. Back then no one hurrah, Gregor Mendel, but he's gone.
Okay, thank you to Moxie Fruvis for that song. Okay. So, when the alleles separate or segregate into the gametes, and each gamete gets one, that's how we start our Punnett Square. Each gamete is equally likely to get each allele. Yes?
Does that mean, even though, like, in your DNA, you have alleles that have a dominant gene, you could have a gamete that only has a recessive?
Absolutely. So the question was, if you are heterozygote and you have a dominant allele and a recessive allele, when you make gametes, your gametes are going to have either the dominant or the recessive, one or the other. And just because that allele is dominant doesn't mean that more gametes are going to get that dominant allele or that gamete has any kind of advantage over the recessive one. So that's a good question, because that's a concept that often confuses people. Any more? Okay, so we're going to go ahead and draw a Punnett Square. I gave you an empty one on your handout. We're going to cross a heterozygous round P with another heterozygous round P. This would be like generating an F2. So you've probably done this before. The gametes from this guy could have a big R or a little r. They'd be equally likely to get the dominant allele or the recessive. The other individual in this cross is exactly the same, so you'll put the exact same gametes up here. These are all going to be equally likely, so half of the time this gamete will be produced, half the time it will be this one. To fill out the Punnett Square, you just merge these two gametes, and when we simulate fertilization of the two dominant gametes, we get a homozygous dominant individual. If we use a recessive and a dominant, we get the heterozygotes out. And in one of these four cases, the two recessive gametes will come together and we'll get a homozygous recessive. So the next step is assigning the phenotype to the genotypes. You have the genotype in your Punnett Square, but you need to know how to assign phenotypes because that can change depending on the situation. Green here is what is the dominant phenotype. I would say wild type, that would be a mistake. So green is the dominant phenotype and blue is the recessive phenotype. So 3 out of 4 of this cross will have the dominant phenotype and 1/4 will have the recessive phenotype. And this is what Mendel saw in his F2s with simple Mendelian inheritance. Now, as another example, we're going to go through cystic fibrosis mating. Dr. O'Dowd introduced cystic fibrosis a little bit earlier, but I'll remind you what it is. It's a pretty common disease. It happens in about 1 of 3,000 Caucasians. It's less frequent in African and Asian populations. Patients have a mutation in a gene that encodes for a chloride channel, and when you can't pump the chloride out, your mucus becomes really thick and sticky, and their airways, the cilia, can't move that mucus out, and they tend to get a lot of infections. That leads to chronic infections and airway obstruction that can damage your airways. So people who have the disease cystic fibrosis often die by the age of 30. It's pretty sad. Mutations that cause this disease are recessive. One normal copy is enough to keep you safe. So the heterozygotes have the dominant normal phenotype. So in this case, we're going to do a different kind of mating. We have an individual with a heterozygous genotype and an individual that's homozygous dominant. So the heterozygote here can make 2 kinds of gametes, and will do so with equal frequency. This person has a special name. They're called a carrier. So their phenotype is normal because 1 copy of the gene is enough to make your mucus not so sticky and you're fine. Carriers have no phenotype, but they do have a different genotype from this totally normal person. So the carrier is referring to their genotype. Their phenotype is normal. Now this homozygous dominant person can only make 1 kind of gamete. They can only make gametes with the dominant normal allele. So you can still draw a Punnett Square with 4 squares if you want, or you can just get rid of this, because they can only make 1 kind of gamete. 100% of their gametes are going to be big C dominant. They are the normal person. For this one, they're present half and half, so you can go ahead and fill out your Punnett Square as before, crossing these gametes, and it looks a little bit different than before. In this case, everybody is going to have a normal phenotype. Nobody from this mating will end up, no progeny will end up with the disease, but half of the kids will be carriers. They will have a genotype that makes it possible for them to pass on the cystic fibrosis gene to their kids. So these people are carriers. So you should be able to do any kind of mating like this with a Punnett Square. Any questions? Okay, here's an opportunity for you to try it out. 2 polydactyl cats living in Irvine mate. They managed to escape the coyotes and had a litter of kittens. What phenotype do you predict for the kittens? What frequency of the phenotypes? Go ahead and give it a try. You are welcome to talk to your neighbor if you like. read and tell you guys that most of you are wrong. So talk to your neighbors and feel free to change your answer. Yes. You can figure this out. There is a right answer. Despite the insightful questions I just got, there is a right answer. Okay, I'm going to close it for polling, so just give your best guess. Let's see what you guys thought. So a lot of you guys like C, and C is the right answer. So that's good. Now, it wasn't like that before, so talking to your friends is helpful. That's good. Now, the key thing here is we have to set up our Punnett Square, and so we have to figure out what the genotypes of the parents are. So, what did you guys think the genotype of the parents was? I'm hearing a lot of muttering. One person raised their hand. What did you think? You asked me a question. So you thought that the parents were heterozygous. Did anybody think the parents were homozygous? Yeah, they could have been, right? But the probability that they were was very low because of something I told you earlier in lecture, right? We had people raise their hands. How many polydactyly cats have people seen? Only 6 or 8 people raised their hands. Polydactyly is a rare allele. You probably don't know many polydactyl people either. Lester is the only polydactyl person I've ever met, right? The allele is dominant, but it is rare. So the odds that someone who's polydactyl has 2 of those rare alleles are pretty low. So you can safely assume that both parents are heterozygous. And that's how you get to this answer. So I think that's why this problem is a little bit tricky. In an exam, I will often tell you that the allele is rare or the allele is common. And that information will help you, oftentimes, assign a genotype to individuals in a cross. Okay? Got another one for you guys to try. This one is a woman who has a sibling with CF would like to have children and seeks genetic counseling. So if I had a brother with CF, I know that my kids would be at risk, and I would go see a genetic counselor. This woman is found to carry the delta F508 mutant of the CFTR protein, while her spouse has a different mutant, G542X. So this delta means it's missing. So phenylalanine 508 is missing. It's a very common CF allele. This one is a little bit more rare, but both alleles produce non-functional protein. What proportion of their children are likely to have the disease? Okay, go ahead and give it a guess if you aren't sure. See what you think. Very good. The most popular answer is the right one. The answer is 1/4. People get confused by this, but it's the same question we did before. Okay, both of these people are heterozygotes, and they're going to have one that's homozygous recessive. It's a little bit confusing that they have different alleles, but both alleles are non-functional. So this heterozygote will have CF. They won't have a functional CFTR channel. Yes?
So we could assume that the mother didn't have cystic fibrosis.
Oh, that's a very good question. So we can assume that the father didn't have CF. Yes. It would have been better if I had stated that both parents were normal phenotype. Yes. But if you had to choose the best answer and make an assumption, yes, that would be the right thing to do. That's a good point. Okay. So now we're going to talk about test crosses. A test cross, the reason why you would do it, is to determine the genotype of a dominant phenotype. So here we have this round P, and I told you that round is the dominant phenotype. So we don't know if this P is big R, or big R, little r. Now, the genetics textbook we used in 97 used to use the letter of the recessive allele, so yours says WW, just change it to RR. That was the one place where I forgot to change it. So it could be a homozygous dominant, or it could be a heterozygote. So we're doing our test cross. We use this P here, which is wrinkled, so it has to be homozygous recessive. When you do a test cross, you use a homozygous recessive individual. So if this unknown round P is big R, what are we going to get here? All round P's. And what is their genotype? They are all heterozygous. Right. Very good. If this test subject was a heterozygote, what are we going to get out of our test cross? Half and half. Very good. Half round, half wrinkled. Because half the time, if this one is a het, it will be giving the dominant allele, and half the time it will be giving the recessive. The only allele that can be offered up by this wrinkled P is that recessive allele. And that's the key to a test cross. The key to a test cross is you have to use a homozygous recessive individual. If you're looking at multiple loci, that test cross person, subject, organism, has to be homozygous recessive at every locus. Because you need to be able to see both alleles that are present in the test subject. So the key to doing a test cross is to use a homozygous recessive individual in your cross. Questions? Sometimes people get confused by this. Okay. So the second of Mendel's principles or laws is independent assortment. In this case, he said, in the formation of gametes, the segregation of any pair of alleles is independent of the segregation of other pairs of alleles. We have here a dihybrid, heterozygous at two loci. It has a dominant phenotype. Yellow is dominant, round is dominant. If this P makes gametes, the plant produced by this P, they are equally likely to be all four of these possibilities, right? So you could have this big Y go off into a gamete with a big R. The big Y could go off with a little r. The little y could go with the big R. The little y could go with the little r. All are equally likely possibilities. And that is the principle of independent assortment. The way I keep this straight in my head is I remember that these are pairs of alleles. We're talking about two different loci, and the alleles at those two different loci segregating independently. So independent assortment is two genetic loci. Now the basis for this is, again, meiosis, which Mendel didn't understand, but you do. During metaphase 1 of meiosis, the homologous chromosome pairs line up on the metaphase plate. Now if these two different genes are on different chromosomes, they have an equal probability of lining up with both dads on one side and both moms on the other. So this is color-coded. The blue alleles are from the paternal chromosomes, and the red alleles are from the maternal chromosomes. One possibility would be that the dominant Y you got from dad is on the same side as the dominant R from dad, which means mom's recessive alleles will be on the same side. But it is equally likely that mom's B chromosome will be on the top of this metaphase plate next to dad's A chromosome. And in this case, you would end up assorting them separately. So dad's Y can go with mom's R. And that is the principle of independent assortment. Now, this is a figure from your textbook where they're showing you what doesn't happen, which often confuses people. So this side is not true, does not happen. It could have been that this heterozygous dihybrid F1 here could only give big R and big Y together because they came from dad as a package deal. But that's not true. They separate. They assort independently during the production of gametes. So this side is not true. So I'm blowing up the one that is correct so we can look at it bigger. This is a Punnett Square for a dihybrid cross. It's a dihybrid because it's heterozygous at two different loci. When you cross this is like creating the F2 generation. So these are the F1s. Each is the same genetically, so we're going to get the same gametes on both sides. We just said that each of these four possible gametes is equally likely. So they'll each be present 1/4 of the time. You can fill out the table pretty easily. But the special thing about a dihybrid cross is with simple Mendelian inheritance, you always get a phenotypic ratio of 9:3:3:1. What do those numbers mean? It means that 9 times out of 16, you're going to get an individual that has both dominant phenotypes. 3 times out of 16, you'll get an individual that has one dominant phenotype, or could have the other dominant phenotype. And only 1 time out of 16 will you get an individual that has both recessive phenotypes. So you can do the Punnett Square if that makes sense to you, or you can just remember the phenotypic ratio. But this is a phenotypic ratio, not a genotypic ratio. If you figure out the frequency of each of the genotypes, it comes out differently. Yes? Does the what play a role? Crossing over. That's a good question. So does crossing over play a role in independent assortment? It doesn't, because the two alleles will still separate, right? So you could cross which chromosome it's on, which sister chromatid it's on, but it won't change the result. So crossing over is not necessary for independent assortment, and it doesn't mess it up either. It's a good question. Anything else? Yep.
Does the term dihybrid cross only apply to heterozygous?
So does the term dihybrid cross apply only to heterozygotes? Yes, so the hybrid is going to be a heterozygote, and the di means two, so it's heterozygous at two loci. Dihybrid cross will always mean something like this. Okay, so now we're moving out of the land of pure Mendel, and we're going to talk a little bit about incomplete dominance. So we have here a true breeding red snapdragon. If it self-pollinates for many generations, they will all be red, and a true breeding white snapdragon. When we cross them, we get the F1 generation, and its genotype is heterozygous, just like we saw before with brown and wrinkled. But now it has a different phenotype. And that means that it's intermediate between the two phenotypes. It is an example of incomplete dominance. When you make an F2 generation from these F1s, you'll get the parental genotypes out again. If you draw the Punnett Square for each of these, it'll look exactly the same as it did with the rounded wrinkled example. It's just how you assign the phenotype to the genotype that changes here with incomplete dominance. So now the heterozygotes have a different phenotype than the homozygous dominant or the homozygous recessive. And I provided you with an example of how this might work, a molecular explanation. So maybe the CR allele that makes red flowers makes an active enzyme, so they can turn red. But the CW allele, maybe it's broken, it doesn't work. It either makes no enzyme or an inactive enzyme, so they're white. So this heterozygote would have one working copy and one busted copy, so maybe it has less red pigment and turns out to be pink. So it has an intermediate phenotype. And this is an example of incomplete dominance. The next variation is codominance, and you've probably heard this example before because it's one of the very few examples of codominance. With codominance, both alleles are heard in a phenotype. Both alleles will control the phenotype. The example here is this carbohydrate that's present on the surface of your red blood cells. It could be an A protein that has the A structure, and that is determined by the IA allele. It could be the B protein that has a slightly different shape than the A. That's encoded by the IB allele. Or if your allele makes no carbohydrate at all, it's a little I. And so you have no carbohydrate. Now people are going to have 2 of each of these alleles, so these are the possibilities. If you have 2 of the A, or an A and a non-functional, you'll have only A protein on the surface of your red cells, you'll be blood type A. Same as with B, except you'll have B on the surface. If you have one of each allele, this is where you can see clearly both alleles. you end up with an AB. You have both proteins on the surface, both carbohydrates on the surface of your red cells. And if you're type O like me, then you don't have any of these carbohydrates on the surface of your cells, and you end up being O. Now, all of these people have a phenotype for receiving a blood transfusion. If you're type A, what kind of blood can you receive? A or O, right? So your immune system is used to A. You won't see A as foreign. So you can get any blood that doesn't have B. If you're type B, you can get any blood that doesn't have A. What about if you're type AB? What kind of blood can you get? Anything. They are the universal recipient, so they are lucky that way. They're not very common. They can get any blood. If you're type O, you need blood that has no carbohydrates because your immune system will recognize any carbohydrate as foreign. Okay, now this slide is very important. I'd like you to pay attention. This is the last slide, so we're almost done. How can two individuals with the same genotype have different phenotypes? These are hydrangea plants. They are clones of each other, and yet they look very different phenotypes. That's because this plant was grown in more alkaline soil. This plant was raised in more acidic soil. As a result, aluminum is differentially available to these plants, and they end up producing different colored pigments. So the key here is that the environment has a profound effect on phenotype. Individuals with the same genotype can turn out different and look different because of differences in their environment. It's critical. Genes in the environment interact to produce your phenotype. Genes at other loci, other genes, are also important in determining a phenotype, and we'll talk about that interaction on Monday. Very few traits, very few phenotypes are determined by a single genetic locus. In most cases, several proteins, several genes are working together. So that's why I made up the favorite ice cream flavor locus, because so few traits are monogenic. All right, have a great weekend. Study for your quiz. Come ask me questions if you need to.